as I was saying, I'll present a bit of light propagation in tissues and how they relate to the different imaging modalities and different approaches that we can use for imaging. Our goal, of course, is to image in vivo. Most of the techniques I will show, they're very useful for for whole organ imaging rather than in vivo, but still some of the techniques can be used for in vivo. And then I'll present the basics of light sheet microscopy. It's a technique that has become very popular in recent years, and for that you need very little scattering, so it really links with this first part of the talk. And then I'll go over to another technique which is called optical projection tomography, which is quite similar to actually to X-ray CT, but in this case using optics. So, related to the basics of light propagation in tissue, we're interested on probing tissue to obtain information. So either you may have a transgenic animal that expresses a fluorescent protein, or you might have a probe that you injected that fluoresces. So we need to understand how light propagates in tissue in order to obtain information from that. So as you know, a tissue has a very complex structure. So depending on the amount of scattering present in tissue, we will need to deal with different types of models. So the important thing is that we need to be able to model how light propagates. Now, in order to get an idea of what happens, you have to understand that the basics of what governs how light propagates, it's going to be, it's full of different, it's like my flight yesterday. There's problems everywhere. Okay, so it's, the most important things when you consider light propagation are absorption and scattering. If somebody gets sick, there's some bags in the back. All right. So depending on the amount of scattering present in the tissue you want to image, you will need to you can use one approach or others, and I'll express this with a specific experiment. This is a very simple experiment. It's just a glass of water where I'm going to be adding some drops of fat. So this is a yogurt. I'm going to be adding drops of fat, so this increases the amount of scattering, and I'll be using a laser pointer like this from the side, and I'm going to be taking photographs with a normal camera from the top. So first, if you don't put any drops of fat, you wouldn't see the laser beam, right? So that's something you should all have in mind. If there's no scattering, you wouldn't see the laser beam because we're looking at 90 degrees, right? Now, if there's only one, then you clearly see that it exponentially decreases as it goes inside. So this is called, this is Beer's law. Probably some of you are familiar with it. But the important thing here is that light doesn't lose its original direction. So we say in this case that light propagates ballistically. This is what happens in microscopy. This means that we can still maintain spatial information when we image, because it's not lost on propagation. Now, the more scattering I put there, the more drops of fat I have there, the more I start losing this original direction, and at the end I end up with this type of image, where the original direction is completely lost. It looks like an isotropic point source at this point, and in this case we say that light diffuses inside the medium. So I'm going to be presenting results that have to do with microscopy. And in this case, we'll be assuming that light propagates ballistically, that we can use microscopy. Now, the next talk, that Joe Culver is going to present, they need to assume that light diffuses inside tissue, or there are other models in between those two that make our modeling of light propagation much better. And this also relates to what Vasilis, I believe, will be presenting at the end, with how you model how light reaches the tissue before you can obtain an acoustic signal. So there are two ways of dealing with this. What I presented is that depending on the amount of scattering, you can measure directly an image, or you need to use some reconstruction for this. So you can separate imaging methods in two types. Direct methods are the ones that it's a photograph. I mean, there's a direct relationship between your measurement and your image, or indirect methods, where you need to do a reconstruction in order to obtain an image. And what I'm showing here is an example of OPT. This looks like a sinogram that you would have in CT imaging. So you need a reconstruction algorithm in the middle. And what I want to point out is that direct methods are really nice because what you see is what you get. But the problem with those is that the more scattering is present, the less information you obtain. So And on the other hand, when you're using indirect methods, it's easier to improve your model so that you can still obtain an image even though there's lots of scattering present. So most of the techniques, the two approaches I will be presenting, one will be for light sheet microscopy, that will be a direct method, and then OPT, which is optical projection tomography, it will be an indirect method, similar to X-ray CT. So in an indirect method, we need to solve what's called an inverse problem. So we need to recover the 3D distribution of fluorescence. This is, for example, what would happen with FMT, with fluorescence molecular tomography. We want to recover the concentration of a fluorescent protein or a fluorescent probe. And for that, we need to solve what's called the inverse problem. So there's a lot of work going on in this, and if anybody's interested, I'll be very happy to provide some reference. So this happens in no matter the amount of scattering. You might need, I will present an indirect method, even though there's very little scattering. There are direct methods also for high scattering. That will be when you use planar imaging, for example, for in vivo imaging, and there you lose a significant amount of information. If you want to recover the 3D distribution of fluorescence, or if you want to recover intrinsic absorption contrast, you will need to use indirect methods like FMT or DOT, diffuse optical tomography, which we will hear in the next talk. So light sheet microscopy is quite simple. Actually, the idea, the first paper published on light sheet microscopy is actually in 1903, so it is, has been around for quite some time. And the idea is very simple: you generate a sheet of light, and you make this sheet of light coincide with the objective of your microscope. I will present this a bit more in detail, but it wasn't until 93 when we see a paper really using this technique. This technique, it was called Opfos then. It didn't catch up, unfortunately, and then we start seeing something similar it's called TSLM 2002-2009. But it's not until 2004 where the group of Stelzer publishes the science paper, and they're using exactly the same idea of that original paper in ultramicroscopy. On ultramicroscopy in 1903, they generate a sheet of light with a cylindrical lens, which, if I take this laser pointer and I put a big pen, for example, which will be more like a cylindrical lens in front, you will see that it generates a sheet of light. I will show this better in the next few slides. Anyway, this gets the ball started, and by 2007, another group publishes something very similar, and they actually call it ultramicroscopy. And in both these cases, we generated a sheet of light with a cylindrical lens. You can generate this same sheet of light using other methods. In this case, they were actually scanning a beam. That would be like scanning this laser up and down, like this, generating this beam, and actually they were introducing special information in the beam, and this increased the resolution. The next step was to use different types of sheets. So to use nonlinear techniques for generating this sheet, and in that case you will get much better resolution. This is already now in 2011, and now in 2012 at the end in December. So this was a very recent paper. There was this paper published using a light sheet technique that actually proved to be much better, much more efficient, and with with lower photo bleaching compared to a spinning disc confocal. So this paper actually brings us into a new era where confocal imaging is being replaced by light sheet microscopy, and this is in vivo in this case. So the idea of spin is very simple. If you generate a sheet of light, the easiest way is if you take your beam, you put a cylindrical lens, this generates a sheet of light, and you make this coincide with your objective. And what you do is you move your sample through this sheet, and you're collecting fluorescence only from that sheet. So the source of contrast in this technique is fluorescence. It's like in confocal imaging, you need fluorescence as a source of contrast. So this is an example. This is an excised lymph node that has been cleared. This is very important. We need to tissue has to be transparent, so we need to clear tissue. It has been cleared and stained for 4 different cell types. What you see here in orange are the HEVs, the high endothelial venules, that look like this once you do have this 3D reconstruction. So with this technique, it's a direct method. You're actually looking at each of the slices, and then you can reconstruct a 3D volume. And I think that's the main reason it has become so popular. And the good thing also is that it has a very low photo bleaching. This is another example more related to in vivo imaging. Important, again, I insist, you need very little scattering. So in this case, this is Drosophila development, where you saw there's a bubble appearing. This is the head aversion process. You will start seeing the head right there with the eyes appear at some point. And in this case, you cannot do SPIM in 3D because there's so much scattering present in the sample that you lose this information. You can still use maximum intensity projection images, at least to obtain a crispier image than what you would obtain with normal epifluorescence. But so in this case, that's why I point out it's slightly scattering. This is another study. This is a mutant Drosophila that doesn't develop the head. So while this head aversion process takes place, we can image it from different angles. You will see at the end that it ends up with basically no head, with a small, very, very, very small, and then they die. These used to develop, they develop normally until they reach this state. So these are just some examples, and here I want to point out again, what happens is that since we have a significant amount of scattering present, this technique is really not good for 3D images, because it is a direct method. So there's nothing we can do. Now, it can be used for several organisms. For example, zebrafish is quite transparent and is being used a lot. It's actually the organism that is being used most with these light sheet microscopy techniques. What you see here, this is a 3D reconstruction of the beating heart of a zebrafish. So we can obtain very high spatial and temporal resolution. And in this case, since it's quite transparent, we can reconstruct this as a 3D image. Now, as it gets older, of course, you have more and more scattering, scales start to appear, and it becomes very difficult to use this technique. Okay, so that technique dealt with the fact that we had very little scattering. And what I want to present now is another approach, very similar. If you want, the equipment you need is very, very similar, but the fact that it needs a reconstruction approach makes it, I think, more versatile. You can use it for cases where scattering is significantly present. So the idea of optical projection tomography, the concept is very, very similar to X-ray CT. The first time it appeared, it was in 2002. It's a paper by James Sharp, and the idea is, as you would have in X-ray CT, where you have your CT source and your detector rotating. What you would have is a normal microscope, and your sample, instead of being placed on your cover slip, you have to mount it on something that rotates, and then for each of the angles you would measure fluorescence. Now, one of the advantages that this technique has is that you can also measure absorption, like you would do with X-ray CT. So it offers contrast comes from both absorption and fluorescence. And the way this works, for those of you that are not familiar, is this should be a movie anyway. You can imagine this rotating. What you collect is, in the case of absorption, you will collect the shadow of each of the angles, and then if assuming, let's say, this is your CCD image, if you take a line of this image and plot that for each of the angles, what you construct is what's called a sinogram. So here you have that line, and these are the angles there. So once you have this information, you can use what's called a Radon Transform, and this gives you an image only of that slice. So by selecting the different lines of pixels, I can then reconstruct a 3D volume. Now, this is that same fly. It's a fly that expresses GFP everywhere. What you're seeing again is this head aversion process. You will start seeing a bubble appear. These are the salivary glands and the fat bodies you can see there. And then at some point, you will see that there's lots happening. So there's not much happening in the first 12 hours or so, and then suddenly in like 20 minutes, everything happens, and you start seeing, now you see the eyes there. You can start seeing you see the legs, the wings. So we obtain this what I'm showing here. This is there's an OPT session, so there's 500 measurements, angular measurements I think we did this every 5 minutes. So what each of these frames represents is a 3D reconstruction using Radon Transform. So that's another thing to point out. These techniques, they generate tons and tons of data. So there's a lot of work going on how to effectively manage all this data. And I don't know if you noticed, but one of the important things is that in one of the frames you saw lots of, suddenly everything was blurry. That's because the pupa is moving so fast the fly in there is moving so fast that when you do one single rotation, the end point and the start point are very different. So those are things that we have to account for, and you can account for in the reconstruction process. So this had to do, so far I went from very low scattering when you can use light sheet microscopy. This technique you can use when there's still significant scattering, because you can always improve on that approach by improving your reconstruction method. And then finally, I like to show something that we're using right now for both for high throughput and for imaging very long samples, which we call the Helical OPT. The idea is exactly the same to what I showed before, with the difference now that very similar to spiral CT, the sample is rotating and then moving upwards. So when you use this, you can imagine, I think I have here some raw data to show you. Imagine this is the OPT I just presented. That will be a sample, will be rotating there, and then for each of these lines and all the angles, I reconstruct what I mentioned before is the sinogram. And with this sinogram, you will get your image in that single slice. Now, in the case of spiral CT, and it's equivalent here in helical OPT, what you have is your sample moving upwards. So you have to, this line of pixels that we're taking, you have to move it together with this vertical displacement. And what you're seeing here, this is raw data, so we can use this for high throughput in vivo imaging in the case of Drosophila, or you can use this also for organ imaging. So you can see here that all these different flies that express GFP as they rotate and they move upward. So then, using this approach, you can image also very, very long samples. We have been used this to image a colon, the complete colon, is 5 centimeters. So we can obtain a low resolution 3D image using this technique. And then, since the equipment is very similar to what I presented before, SPIM, to any light sheet microscopy method, you can use SPIM to obtain a higher resolution image in those areas that you're really interested in. So what you're seeing here, this is colon tissue. These are 3 slices here with these three green arrows. These beam images are from this blocks. And what you see here are pair patches from another colon sample. So our idea is to use, when you want to image long organs, is to use helical OPT to obtain a lower resolution 3D image of the whole organ, and then select those areas and obtain a higher resolution image using SPIM. Again, I insist we can use this because scattering is really, really low. So there's a lot of work going on taking organs and clearing them. So you put them, for example, in this case, we put them in a solution called BABB, Murray Clear, which is very aggressive. And what it does, it basically destroys all proteins and it becomes transparent. So if you have a fluorescent protein that you're interested in imaging, it also destroys this. So you need to use either fluorochromes or you need to resort to other clearing techniques. And there have been several published. One is Clarity, has been published recently. Clear-T is another one. Scale is another one. So there's lots of publication coming out in recent years showing different methods that will still maintain the fluorescent properties of your sample. So as I said before, we can use this also for high throughput. What you see here, we had 13 of these flies stacked up together, and we can obtain 3D images of each one of them. What you're seeing here, this again are the salivary glands. You start seeing this bubble appearing here, and we can also see the header version. So we can do high throughput time-lapse imaging on these specimens. So with that, I like to close. Hopefully, I conveyed the point that scattering will tell you, actually limits what type of approach you can use if you want to obtain the maximum information possible from your system. So there are 2 methods, at least that's the way I separated it. They have direct and indirect methods. Direct methods, they're simple, and what you see is what you get. So you take a single photograph of a slice, and that's your image. Indirect methods, they have different levels of complexity. I think that's the reason they're not so commonly used. FMT, for example, is one of these methods, but they are flexible. So if you account for light propagation in complex media, you can still obtain your 3D image. So light sheet microscopy and optical projection tomography, they complement each other very, very well, because they use different optics. So you can obtain low resolution images of large volumes and account for light propagation, whereas with the light sheet microscopy with SPIM, you can obtain high resolution images within the sample. And with that, I'd like to finish. And if you have any questions, I think we have maybe a few minutes. Well, thanks.